Hello students, today we are going to start the topic of translation from chapter 6 Molecular Basis of Inheritance. To start the topic translation, I introduce the central dogma of protein synthesis. We already know DNA, transcription, messenger RNA, translation, protein. We have covered this part, transcription, in which the sequence of the DNA is coded on the messenger RNA. And here is the messenger RNA. On the messenger RNA, genetic information is present in the form of genetic code. And this code is decoded in the form of the protein. Here we have sequence of nucleotides. This will govern and the result will be chain of amino acids, or we can say a polypeptide chain according to the sequence of the genetic code present over the messenger RNA. So here we are showing the genetic information and this is the result, and this pertains to the concept of translation. This is our today's concern. To start translation, we need molecules of RNA, rRNA, tRNA, mRNA. rRNA, ribosomal RNA, it forms about 80% of the total cellular RNA and is a component of the ribosomes. We know that RNA present in the ribosomes is called rRNA. It is a single-stranded molecule but twisted on itself. What is its function? It is the site during the protein synthesis, and it contains some proteins called ribosomal proteins and rRNA. Second is messenger RNA, mRNA. It is formed by DNA template in the nucleus and moves to the cytoplasm within the two subunits of ribosomes. This we will study when we proceed toward translation. It is a complementary strand to one of the DNA strands formed during transcription. It forms about 5 to 10% of total RNA in a cell. So this was the major percentage, this is a small percentage. Its length is almost equivalent to the length of protein to be synthesized in the cytoplasm, because this is the chain from where the protein is going to be synthesized. It has a cap structure at 5' end and a poly A tail at the 3' end. It's both the ends are defined, 5' end and 3' end. Its main function is that it has genetic information. It carries codons which serve as a message tape to be decoded into a protein. Protein means chain of amino acids. Next we proceed to transfer RNA, tRNA. It is the smallest of all the RNAs with molecular weight ranging from 25 to 30,000 Daltons. It is soluble RNA and constitutes about 10 to 12% of total RNA in a cell cytoplasm. So this is the smallest and this is the soluble type of RNA. Its function is like an adapter. It picks up activated amino acids from the cytoplasm and supplies it to messenger RNA in ribosome according to the message expressed in the form of the codon. The codon was there on the messenger RNA, and it picks up the concerned amino acid and brings it at the site of the codon. Each amino acid bears the following sites: recognition site, anticodon site, ribosome attachment site, and amino acid attachment site. This will be clear when we proceed to the diagram of transfer RNA. Further, Francis Crick proposed the presence of an adapter molecule which could read the code on one end and on the other end would bind to the specific amino acid. This is the property as an adapter. However, transfer RNA was known before the genetic code was postulated and was then called sRNA, soluble RNA. But its role as an adapter molecule was reported later. Now, we proceed toward the structure of the transfer RNA. Here is the diagram of the transfer RNA. The secondary structure of transfer RNA is clover leaf-like. It is also, in a different view, it gives L-shaped molecular structure. But here we are showing the clover leaf structure. Transfer RNA has 5 arms or loops. We can see these loops. Anticodon loop, which has bases complement to the codon. Here is the anticodon loop. This is the anticodon loop, and these are the anticodons, which are just parallel to the codon on the messenger RNA. Amino acid acceptor end, to which the amino acid binds. Here, this is the amino acid accepting site, where the amino acid attaches to the transfer RNA. Tail loop, which helps in binding to the ribosome. Here, it binds with the ribosome with this tail loop. D, which helps in binding amino acyl synthetase. Amino acyl synthetase is the enzyme, and here this D loop helps to bind with the enzyme, which is involved in protein synthesis, that is amino acyl synthetase. Then we have variable loop shown over here. Now, some more properties about the transfer RNA. Each transfer RNA is specific for a particular amino acid. Means to bring a particular amino acid, a particular transfer RNA is involved. A specific transfer RNA for initiation is called initiator transfer RNA. But to start, there is a specific transfer RNA every time to start the genetic code or the information on the messenger RNA. There is a particular codon and there is a particular transfer RNA for it. That is called initiator transfer RNA. Actually, this is the one which will carry methionine. There is no transfer RNA for the stop codons. This is another important feature. When the chain on the messenger RNA shows stop codons, there will be no transfer RNA for them. That is how the chain stops, and we will study it later as we proceed toward translation. With this, we have just covered 3 types of RNAs: rRNA, messenger RNA, and transfer RNA. All the 3 are very important to play the role of translation. Apart from it, we need some energy molecules, some enzymes, and let us proceed toward the topic of translation. In translation, there are 3 stages of protein synthesis. Broadly speaking, there is initiation, elongation, and termination. Here is a brief outline about these. During its initiation, there is assembly of the ribosomes on the messenger RNA. Also, the activated amino acid and its delivery to the transfer RNA. It means the transfer RNA will bind to the amino acid and finally bind to the messenger RNA. It will be more clear when we proceed to the diagram. During elongation phase, the repeated cycle of amino acid delivery is there. It means more and more amino acids are delivered with the help of transfer RNAs. Peptide bond formation, because between the adjacent transfer RNAs, the peptide bond will be formed, and movement along the messenger RNA is called translocation. Again, it will be clear when we proceed with the help of diagram. And in the termination step, there is release of a polypeptide chain. This is the final result. Then other components get dissociated and there is a release of the polypeptide chain. Now we start with initiation. Let's proceed to the diagram. We start with initiation. For this initiation, the smaller subunit of the ribosome binds to the messenger RNA and it binds to near the 5' end, and this is the first transfer RNA, which is methionine transfer RNA, but in prokaryotes F is added for methionine transfer RNA, and it comes and binds with the first codon, which is always AUG, and here it has reached, and at this time the second subunit will bind. For this, some initiation factors are also required, and when the first transfer RNA has reached this codon, the step of initiation is there. Now I explain further. Before we reach this, that transfer RNA comes and attaches over here. The amino acid is activated, and activated amino acid is added to the transfer RNA. I show you with the help of some reactions. First step is activation of amino acid. The amino acids become activated by binding with amino acyl transfer RNA synthetase enzyme in the presence of ATP. Basically, it is amino acid plus ATP, and this is in the presence of enzyme amino acyl transfer RNA synthetase. What happens? AMP enzyme complex. It means enzyme is also attached. AMP is attached from the ATP. It is adenosine triphosphate, it is adenosine monophosphate, and extra phosphate is independent phosphate is over here. Now this is the charged amino acid, or we should say rather activated amino acid. When this activated amino acid is added to the transfer RNA, and this we study in the second step, transfer of amino acid to the transfer RNA. Now this activated amino acid, AMP enzyme complex formed, reacts with the specific transfer RNA to form amino acyl transfer RNA complex. This is how the activated amino acid is binding with the transfer RNA, and we get amino acid transfer RNA or amino acyl transfer RNA plus AMP plus enzyme. AMP is free, enzyme is free, and here is the transfer RNA carrying the amino acid. These steps are essential before they reach the messenger RNA. The cap region of the messenger RNA binds to the smaller subunit of its ribosome. The ribosome has two sites, A site and P site. The smaller subunit first binds to the initiator messenger RNA, then binds to the larger subunit, so that initiation codon AUG lies on the P site. The initiation transfer RNA, methyl transfer RNA, then binds to the P site. We again proceed to the diagram. Here is the transfer RNA with which methionine has been attached, and we have studied this step. First the amino acid was activated, then it attaches with the transfer RNA, and such a transfer RNA is charged transfer RNA, and it will attach with the first codon AUG, and which is near the 5' end. Already at this stage the ribosome smaller unit has bound to the messenger RNA, then this transfer RNA which is charged and binds to the first codon AUG. Here again we are showing, and after this the second subunit also binds. Another important point which we discussed earlier, that this ribosome has two sites, A site and P site, and the first transfer RNA enters in such a way that it attaches at the P site. So the ribosome has two subunits, smaller and larger, and in this unit there are two sites, A site and P site. The transfer RNA binds at the P site, and this step is initiation step. We proceed to elongation. During this elongation, another charged amino acyl transfer RNA complex binds to the A site. Here this is the second one. It binds to the A site of the ribosome at the second codon. Now it is binding at the second codon. This one comes from the A site. A peptide bond is formed between the carboxyl group COOH of amino acid at P site and amino group NH of the amino acid at A site with the help of enzyme peptidyl transferase. Now is the time when peptide bond is formed. A peptide bond is formed between this and this. So, the carboxylic group of the amino acid at P site, NH group of the amino acid at A site. This is, we have already studied when a peptide bond is formed between the two amino acids. And when this bond is formed, this transfer RNA will be free. We can see it over here. This was valine over here. This is methionine, the first one. So the second one is carrying both the amino acids. A dipeptide has been formed. And this is the one which is going out. Released transfer RNA. This is its outward passage. It is going out. When this happens, you see a change. Here this valine, the transfer RNA carrying the valine was located at the A site. And in this picture you see that the transfer RNA carrying the valine is located at the P site. How does it happen? During this process, the ribosome moves in the 5'3' direction, which is called translocation. This is how the one which was at A site now shifts to the P site, and A site is again vacant for the newly coming transfer RNA, which is already charged, means which is carrying the activated amino acid. So, the ribosome slides over the messenger RNA from codon to codon, from one codon to next codon to codon in the 5'3' direction. According to the sequence of the codons, amino acids are attached like this. According to the sequence, new amino acids are brought and attached in this peptide chain, and a peptide chain is formed. They are brought with the help of their respective transfer RNAs, and a peptide bond will be formed from the newly coming and from the previous. At one time there will be only 2 transfer RNAs during this process. Here this is the outgoing and this is actually coming at the A site. Now we proceed to termination. We have already discussed that the ribosome keeps on moving from codon to another codon, and this is how every time we get the previous one at the P site and A site becomes vacant. And when the ribosome is moving, especially when A site of the ribosome reaches a termination codon, finally a stage comes when this A site reaches a termination codon. We have already studied stop codons. I'm using the word termination codon or a stop codon. We have studied in the genetic code. If it reaches at that place, which does not code for any amino acid, because that stop codon does not code for any amino acid, no charge transfer RNA binds at A site, and then there will be no incoming transfer RNA, and the site will remain vacant. Then what happens? Dissociation of polypeptide from ribosome takes place. Everything dissociates. You can see the subunits of ribosome have dissociated, smaller unit, larger unit, which is catalyzed by the release factors. There are some release factors which help in this dissociation. There are 3 terminator codons or the stop codons, UGA on this, or UAG or UAA. When they are at towards the 3' end, anywhere towards the 3' end, there will be no further transfer RNA which can bring any amino acid, because there is no transfer RNA for these codons and no amino acid can be brought. And this is how the growth of the chain stops. Not only the growth of the chain stops, all the machinery dissociates. Messenger RNA is free, larger unit is free, smaller unit is free, and this was the last transfer RNA. It is also free and it releases the polypeptide chain. Here you further see. So students, we have seen the process was started with initiation where the amino acid was activated. Then it was added to the transfer RNA. Then this transfer RNA joins with the messenger RNA towards the 5' end. And ribosome we have seen it has A site and P site. And this newly coming messenger RNA joins at the P site and it fits in such a manner on the messenger RNA that start codon is over there, which is AUG, and here the first transfer RNA comes, which is always methionine transfer RNA, and if they are prokaryotes it will be formyl methionine transfer RNA. And with this step, when the first transfer RNA binds, the initiation is there and it binds at the P site. Remember, the first one comes from the P site, but the new one will come from the A site. During elongation of the peptide chain, another charged amino acyl transfer RNA complex will bind at A site. What is this amino acyl transfer RNA complex? It is the transfer RNA with activated amino acid. It comes and binds at the A site because we have seen in the diagram A site was vacant. It comes and binds at this, and now a peptide bond will be formed between the two. Again we proceed to the diagram. Yes, when the second one has entered from the A site, the earlier one was at the P site. A bond is formed between the second and the first amino acid. So, the second one will carry both. First it will carry valine, then methionine. It is clear from here. It is carrying valine directly and it is carrying methionine because the transfer RNA, the earlier one, has handed over its methionine to the second one and it is being released. And now when the ribosome moves in the direction 5'3', this is important step every time because how to read the new codon? It is the movement of the ribosome called translocation which reaches to the next codon, and this is how, due to the movement of the ribosome, the one which was at the A site is now at the P site and A site is vacant, and this we can see the newly coming amino acid entering at this. This is how the chain keeps on growing because again it will be in this condition and they will bind. Now valine, methionine and then phenylalanine will be bound, and again this one will be freed and this will carry the 3, and like this the chain keeps on growing. Now we proceed back to during termination of polypeptide, when the A site of ribosome reaches a termination codon which does not code for any amino acid. We know the stop codon does not code for any amino acid. No charge transfer RNA binds to the A site. Now there is no transfer RNA to bind, and so no amino acid will be brought. And then what happens? Dissociation of polypeptide from the ribosome takes place. The chain of amino acid is polypeptide and it dissociates from the ribosome, which is catalyzed with the help of release factors. So the release factors help in the dissociation process. When it happens when there are stop codons and no more transfer RNA will come. Release factors will operate and the polypeptide chain is released. So the release of the polypeptide chain, which is actually a protein chain in the cytoplasm, is the result which was there on the DNA and transcribed on the messenger RNA and finally decoded in the form of the protein, which is actually a polypeptide chain. And this is here the translation is completed. So we again move to the diagram of this termination. Here we can see the release of amino acids and the dissociation of all other components. Ribosome larger unit, ribosome smaller unit, messenger RNA, release factor, transfer RNA. Everything has dissociated and the chain has been released and the polypeptide chain formation is clear. This is how the translation is completed.